Nature is nothing if not colorful. Everywhere we look, we're greeted with the millions of colors of the biological world, and most fascinating of all is the incredible diversity of the way those colors are produced in living organisms. While there exist many small molecule dyes like anthocyanin or keratins, not all organisms rely on complex chemical pathways to make pigments. Instead, many forgo these pathways altogether and instead use colorful proteins, which, while more complex, are far easier for cells to produce. There are examples of this everywhere, but some of the most famous come from the oceans. If you've spent any time learning about biology, you'll have almost certainly come across GFP, or green fluorescent protein. We actually talked about it in a previous video. As color proteins go, GFP is about as ubiquitous as they come. It was the first to be discovered and harnessed, and originally came from a species of jellyfish. These days, it's used in a massive variety of biological experiments, anytime you need to visually track something. You can add it to proteins to make them fluoresce, or just add it to cells to see which ones have been modified. GFP is what's called a beta-barrel protein for fairly obvious reasons. At its core is a small cluster of amino acids that do most of the work and are responsible for the glow when it's exposed to UV light. But if you change these amino acids and tweak the structure of the beta-barrel slightly, you can shift the color from greenish to almost any other color. These days you can get fluorescent proteins in a literal rainbow of colors, some even fluorescent in the infrared range. And they don't even need to be fluorescent either. Some varieties don't glow under UV, but instead are just brightly colored in white light, so their utility as pigments is exceptionally broad. Personally, I'm not actually a big fan of GFP and prefer RFP, or red fluorescent protein, more. It's easier to see when a cell is red than yellow, as most cells already look kinda yellow to start with. RFP, and specifically the variety I have, is also natural in origin, though in this case, rather than a jellyfish, it comes from an anemone. What I find neat is that rainbow I just showed you actually just started with these two proteins. Researchers simply mutated both fairly randomly until new colors showed up, and then tweaked as necessary. That's not to say you can't find beta barrels in nature in almost any color, but you can make them artificially or tweak natural ones to be more vibrant, but we'll talk more about that at the end of the video. Last week you saw me build this awesome bioreactor lantern, and as a first test I inoculated it with some RFP producing E. coli. While that was a test to make sure the lantern worked, it was also for another purpose. I've been wondering for a while if fluorescent proteins can work as a dye for a simple glowstick reaction. I'm not expecting it to be good or last very long, as these proteins are pretty sensitive to the hydrogen peroxide used in glowsticks, but I do want to see if there's at least a dim glow for a second. The plan is to process all of this down into some purified RFP to test the glowstick chemistry on. Once that's done, I'll be sending off a sample to none other than Nurdrage himself to give this a test and see if it's possible to be used in this way. This is also a good excuse to go through some very basic protein extraction techniques, and show how you go from growth media to final product. So let's jump right in. The process is actually really simple and is made of 3 basic steps. First, we collect all the E. coli cells and get rid of all the leftover LB broth. Then, we suspend the cells in a bit of distilled water and break them all open. Finally, we process this crude mixture to get rid of all the cell debris and collect our protein. To get started, using the fluid output port of the bioreactor and a syringe, I collected about 10 milliliters of liquid at a time and transferred it to 6 15 milliliter Falcon tubes. I made sure to get the amount of liquid as close to even as possible so the centrifuge will be balanced. This becomes more and more important the faster you plan on spinning the tubes, so for later steps that need to go faster, I took more time and a scale to make sure this was perfect. I'd spin the tubes down for a few minutes to collect the cells, pour off the top liquid called the supernatant, and then add more broth on top before spinning it down again to repeat the cycle. This would be much easier with a bigger centrifuge, but you work with what you have. Once that's done, I added a bit of distilled water to each tube and resuspended the cell pellet and condensed everything down to 3 even tubes. This is to make it a little easier to handle and also to keep several samples to give me room to adjust the protocol, as the first time I tried this I used my entire stock and made one mistake at the very end that ruined the entire batch and denatured my protein. Now it's time to break open the cells and release the proteins. There are lots of ways to do this, and you can use whatever method you're comfortable with. You can also buy lots of great kits tuned to whatever organism you happen to be working with. At first I tried using an enzyme called lysozyme, but that didn't really do much, which was rather disappointing. I probably messed something up with that procedure though, so I'll revisit enzymatic lysis in the future. Instead, I chose to use the ultrasonic homogenizer that we built a few weeks ago, as this is literally what it was designed to do. It's nice because it's a chemical-free method and relatively easy, if a bit slow. The one downside is the probe gets pretty warm, so I can't leave it running for as long as I'd have liked, and have to cool it down between runs. This is why it would be nicer to have a stronger probe like the kind you can buy. It gets the job faster without heating your solution. But this still works very well. To make this easier, I transferred the cell suspension to smaller 1.5 ml tubes so the probe can easily reach into the liquid. After about a minute to a minute and a half of sonication, I'd spin down the tubes, which collected all the broken bits at the bottom and leaves the protein floating in solution. This solution was collected in a separate tube. Unfortunately, one downside with sonication is it's prone to do a great job of suspending bits of cells in solution, so it can take a while to spin it for long enough that the solution fully clears. Don't worry too much about this, as we'll do some cleanup steps after. The process is then repeated over and over again to process the rest of the cells. As this goes on, it's interesting to note that the pellet gets progressively paler as more of the RFP is liberated. Once all the cells have been lysed and you've got a crude protein solution, it's on to cleanup and purification. To get rid of any remaining cells, I used a syringe filter with 0.22 micrometer pores that are so small that bacteria and large debris can't get through. The difference is pretty outstanding, and the solution cleared up a lot after this. Now we want to get rid of most of the protein, so to do this we make a concentrated solution of ammonium sulfate and add 1.5 times the volume to our protein extract. This makes the majority of proteins drop out of solution. In theory the RFP should also, but in my case it didn't, which actually turned out to be a good thing. Once we spin this all down, most of the protein chunk collects at the bottom and the RFP is still in solution. The final step is to add some 95% ethanol to the mixture and give it a quick mix and then spin. The RFP will collect at the interface that forms between the ammonium sulfate solution and ethanol, and using a P1000 tip with the end cut off, this can quickly be collected. After repeating the same thing for all of my extract, I was left with this small tube of RFP suspended in ethanol. This will keep it sterile and relatively stable until I need it. Throughout the whole process, I had a UV light with me and would periodically check all the solutions to make sure as much of the RFP had been recovered as possible, and to visualize which parts it was still contained in, be it the various pellets or supernatants. But even then, I could see that a fair bit was left in my cells, and mostly was too lazy to go back and get every last bit. This goes to show why proteins can be so expensive. This vial contains most of the RFP from about 300 milliliters of harvest from the lantern, and there's only a few milligrams here. And this would be considered crude and only the first step for something that would be sold on the market. From here, this suspension would need to be purified further and processed by things like an HPLC to get it up to the standards of an analytical lab. But for my uses, this is more than sufficient. As I said, I'll be sending some of this off to Nurdrage for eventual testing and look forward to the results of that, even though I'm skeptical anything will actually happen. Based on how poorly soluble this is in ethanol, I can't imagine it'll be overly soluble in the solvents in Glowsticks, but I guess we'll find out. Now to close out, I want to come back to that rainbow I talked about at the beginning. Like I said, you can start from GFP and RFP and make a huge rainbow of color proteins. Well, the thing is, most color proteins on the market are locked down pretty tight with material transfer agreements and copyright and such. So what I want to do is make my own set of color proteins. What myself and Vesta will be starting on shortly is a mutation experiment where we start with RFP and see how random mutation affects the colors that are produced. We'll be using manganese PCR to induce mutations in the RFP gene, and over time, this should shift the color, hopefully dramatically. And when we're done, I plan on releasing the genes for anyone to use. But we'll talk more about this project in the coming weeks as we get started and have some results to show off. Maybe by the end I'll do an extraction like this on all of them and do some watercolor painting with all the pigments. Speaking of color pigments, if you want to learn more about them and see some amazing work being done with them, I'd highly recommend checking out my friend Sebastian Kosioba's work. Just like we'll be doing, he started with some natural color proteins and mutated them to be vibrant colors, which he's working to express in plants. Specifically, he's currently working on making a blue rose. So if that sounds interesting, I've put some links to his stuff in the description. And that's where I'll end this episode. As always, a huge thank you to my amazing patrons and channel members who make these videos possible. And if you'd like to see these projects long before they end up in videos, be sure to head over to my other social media pages, especially Instagram. That's all for now, and I'll see you next week.